Welcome back to the course Fundamentals of General Immunology, and we are in module 3, and today we'll discuss about generation of immune receptor diversity. And in the last class we looked into how these immunoglobulins are divided into isotypes and further into their types. What are the characteristic features of the immunoglobulins, how these antigens are defined, and since we also understood that there are variety of antigens which are there in the environment, there is a need of that many repertoires of antibody in order to fight with those antigens, and therefore always in body we encounter that there could be a multiple ways to make this variation in the antibodies so that these antibodies can bind very efficiently with an antigen to nullify or clear it from the body system. So let's try to understand this antibody diversity, and in this picture which we can see here, so this is actually immunoglobulins which we have discussed in the last class, and essentially these are present on the surface of a B cell. So these are a kind of a B cell, so this is a B cell, and these are present on the surface of B cell. So these are a receptor on the B cell, and if we closely look into these variable region which are there in the immunoglobulins, we could say that there could be an hypervariable region 1, 2, 3, and also these are otherwise called as complementarity determining regions. They are named like CDR. So hypervariable regions or CDR are the same way. So we could just say that HV is what similar to CDR. Now these variation in the diversity of the antibodies start right from the very early stages of development. So diversity in the specificity of antibodies is initially generated at the earliest stages of B cell development in bone marrow. So in the bone marrow we see that there could be a progenitor cells. So the progenitor cells are the mother cell, and from these mother cells we could see that there are multiple daughter cells are coming out which can do a functionality and do some say downstream activation pathways and all. So in bone marrow these progenitor cells are matured, and these progenitor cells are having a gene, typical gene which can code for an immunoglobulins, and we can learn it in a detail because there is a rearrangement of the genes which we need to understand randomly in the B cell. So these B cells are having a gene which can code for immunoglobulins, both heavy and light chains. So we can see that these are the heavy chains, these two are heavy chains, and these two are the light chains here. So there are genes in the B cell which can code for these proteins, and these proteins are randomly arranged in order to make a variation in the chain, and therefore we can see a different repertoires or different kinds of immunoglobulin altogether because of this gene rearrangement. So we have to carefully look into this rearrangement of B cell which can lead to the variation in the immunoglobulins. All right. So the antibody diversity which we are going to discuss in details is actually depend upon the three basic stages. The first stage is, as I discussed, gene rearrangement. So there could be a rearrangement of the genes which we will study in detail. So what we know that once there is a replication, so suppose this is a nucleus, and the nucleus we can see that there are DNA materials, and then these DNA can do a transcription to form this messenger RNA, and these messenger RNA is actually carrying all the information for protein synthesis. Now these messenger RNA once coming into the cytoplasm, just before coming into the cytoplasm, these messenger RNA which are produced in the nucleus are rearranged in order to make a functional RNA. What do you mean by that is, suppose there are this is a pre-mRNA. So the pre-mRNA, might have genes which are called exon 1, then it will be intron 1, intron 1, then we could have exon 2, then we could have intron 2, like that. So once there is a splicing, the phenomena is called splicing, and in splicing the genes are getting rearranged, and finally what we are getting is actually the exonic sequence which can code for a mature mRNA that can code for protein. So essentially these messenger RNA which are matured is coming out. Now this splicing phenomena is important, and because this E1, E2, arrangement of I1, I2, this intron and exons are very important, and this is what we will study in gene rearrangement, because gene rearrangement can lead to variation in E1, E2, I1, I2 and all, which could lead to a very different variant of mRNA, which we will see in little detail. Second is diversity by an enzyme called TDT, terminal deoxynucleotide transferases. These are again very specific enzymes which can do a specific addition of nucleotide at the 3' end of a single strand DNA molecules. So we'll see the way how it does in DNA. And then finally we could look into the somatic hypermutations. Somatic stands for cell, soma means cell, hypermutation means a varied form of mutation, a multiple kind of mutation which are there in the cellular system. So these are the 3 stages which we'll discuss that could lead to generate variety of different antibodies in the human body. Gene rearrangement, the terminal deoxynucleotide transferase activity, and somatic hypermutations. Okay. So let's move on to gene rearrangement, and as I said that there is a phenomena of splicing, and we need to understand few terms here which is critical. So there are 3 segments, rather 4 segments which are present in these heavy and light chain. So the heavy chains which can code for the longer arm of the immunoglobulins are having 4 segments. For example, the V variable segment, D segments, J segments, and then constant region of the exons. So essentially we have variation of V, D and J, and in light chain we don't see D. So D is absent. So just keep in mind that D segments are not present in light chain. We only have 2 variation in the L light chain. We called it kappa and lambda, which we have discussed in the last class, and in heavy chain we have 5 variation which can lead to 5 different kind of immunoglobulins like IgG, IgM, IgA, IgD, and finally IgE. So these are the 5 variations in the heavy chain that could lead to production of different immunoglobulins. Now these heavy chains are having, as I said, 3 segments in the variable domain, that's called V segment, D segment and J segment, while in case of light chain, which is either kappa or lambda, we are not having D segment. Now what is happening that there is a somatic process in B and T cell that combines different regions of the gene altogether. So if we see closely that there are multiple segments of the V here, there are multiple segments of D here, there are multiple segments of J here. Now there could be a possibility that this can bind to this and this. There could be another possibility that this could bind to this and this. That means there are multiple combinations which are possible when we see that how these things are happening in the germline. So basically this is a gene segment. There could be a recombination in the level of DNA where D and G recombination are happening in the first instance, and then V comes into the picture. So ultimately what is the meaning here is the genes are having different segments, and out of all these combination, one successful combination can make a very viable antibody or immunoglobulin, which stands for the variation in the immunoglobulin chain will depend upon how many segments are there in the variable region. And therefore, we could see that this DJ recombination and finally VDJ recombination is giving a unique pattern to the immunoglobulins. So, the variation is happening because the process is completely random, and because of the randomization, we can see variation in the immunoglobulin. So, let us see it in a more detailed way so that we can understand what do you mean by gene rearrangement. So, just keep in mind that there are so this is what we are trying to discuss here. So, this is a constant region, right, and this is a variable region. So, this is variable and this is constant region, right. Similarly, we have a variable region and the constant region in the light chain as well, right. So, we are mostly trying to understand this variable region here. So, these are all mutations which are there in the variable region, while these are the region which are for the constant region. So, let us try to see it in more detail. So, here we are trying to see how the light chain is arranged. So, in the germline, so these are the DNA which is present in the nucleus of our cell. So, we have a constant region here, and then we have a V and J region. Remember, we say that we do not have D in the light chain. And these are the leader sequences. So, leader sequences stands for the promoter sequence in the gene, which can give docking to the different transcription factor in order to start the transcription of the gene. And therefore, these are also important. But our concentration here is towards the variable region and the J chain, J segment. So, there are various type of fragment which we have discussed. So, these D and J cause a recombination. Of course, this is not applicable for the light chain. We will see it in a heavy chain aspect, but here we can see that V and J recombinations are happening in the sequence. So, V and J can bind here. For example, say here we are saying that there could be a 30 to 38 fragments of the variable possibility, and in J there could be 1 to 5. This is just approximation. Say for example, L V could be 30, V the 30th fragment is coming, and V 30th fragment is coming, and in J 4th fragment is coming. So, what will happen here? We will get V30 J4. There could be another possibility that V28 will come with J3. So, in that case we will get V28 J3. That means there could be multiple variation that is possible here, and these are happening in the level of DNA because these are somatic recombination. Once these recombinations are happening, we will get the primary transcript after transcription. So, this could be a primary transcript, and we could understand that these are all exons. So, this is also exon, this is also exon, this is also exon, while in between we have an unwanted region we call introns, and this will be spliced off during the process of splicing. So, exactly after splicing, what we are getting is a matured form of the messenger RNA which is ready to form the protein. So, what is happening? The introns are out, and only we could see that these introns are exons are bound. So, what we will see here is all the introns are out and exons can bind to form a functional messenger RNA which could lead to the protein production. Now, we can see that there is a variation here in the hypervariable region, or what we understood, this is called complementarity determining region of the immunoglobulins chain. And since this is light chain, so we are trying to focus here in the light chain. Now, we could see that these variation can possibly various type based on what kind of combinations are coming from here, right. So, we will try to see another one here in the heavy chain exactly in the same fashion. So, we have a constant region here, and we have here an additional region. So, diversity in the heavy chain is more as compared to light chain because we have a D segment additionally here in addition to V and J. So, we have approximately 40 segments of V. There are 23 segments of D. This is just a kind of pictorial representation to give you an understanding of this gene rearrangement, and J is around 6 in number. Suppose if there is a first is, that is what our term coming from here, D and J rearrangement. So D and J will combine first. So 23 segments of D and 6 segments of J will combine randomly. For example, let's say D is 5th in number, so D5, and say J6 is coming here. So ultimately it is having 5 and 6 combination, 56 say for example. And here V is as such, and the next round we are getting VDJ recombination. So DJ is done, V is coming. So now V is 14 numbers. So maybe here we could say that V is 35 number, V is doing an arrangement with D5 and J6. So ultimately we could get a variation in the VDJ chain, which is having very specifically 35 fragment of the V, 5th fragment of the D, and 6th fragment of the J. So this will make a variation in the immunoglobulin chain, while constant region is here. Now this will do exactly the same what we have studied in case of heavy light chain. So we could see here that ultimately these introns will be out, and finally we get a very concrete form of functional messenger RNA, which could lead to the production of protein. Now here we can see that this is what is there. So these are the heavy chain. So this CH1, CH2 and CH3 is coming from this particular area, and the variation is coming in VDJ chain here. So these are on the top. So there could be a variation in the VDJ chain that could happen here. Now in this case we are saying that this is 35, 5 and 6. These are the combination, and there could be multiple possibility that can happen in the heavy chain as well. So what it indicates that these two phenomena of gene rearrangement in the heavy and light chain respectively can produce variety of different structure on the CDR that can bind to a variety of antigens. So there could be a possibility, and this is what is very unique thing about B cell. We don't see this kind of phenomena anywhere else in the human body. So this is very unique to the B cell lineage in the bone marrow, exactly in the time of maturation. Let's see a little ahead of it, and we can just try to analyze it empirically and see that how things are happening. So we have discussed this phenomena again just to highlight how many different possibilities are there. So we can say that, there are 51 and there are 5 here, and so V and J recombination can lead to say 51 into 5 different combinations. And we could also see that there could be a possibility that there are 65 into 27 into 6 combinations that could be possible in a heavy chain. And finally, when we rearrange it, so we can say that there could be a possibility of 51 into 5 into 65 into 27 into 6. So one can understand, this can just give you an idea the variation and different kinds of possibilities which can lead to the combination, and how many different varieties of immunoglobulin can be produced using this algorithm. So this is a very unique phenomena that's happening in the B cell that can lead to the gene rearrangement and variation in the immunoglobulin chain. So this is very critical in order to produce different repertoires of immunoglobulins in the human body. Let's try to move on and see the second phenomena, and that's basically an antibody diversity based on the very specific enzyme called terminal deoxynucleotide transferase, TDT. Now this terminal, as name itself indicates, it's in the at the corner, terminal at the end. Deoxynucleotide, it stands for, it is very specific for DNA segment. And transferase means it can transfer something. We'll see what it can transfer. So this is an enzyme. This is just a cartoon of this enzyme, and we can see that this is a small single-stranded DNA with 3 nucleotides here, A, C, and T. And in the 3' end we have a hydroxyl group, and the 5' end you have a phosphate group. Now, since there is terminal deoxynucleotide transferase, will add a very specific moiety at the 3' end of the growing chain of a DNA, with the help of divalent metallions. So divalent metallions can be a cofactor for this whole process, and these are all nucleotides which are there. So we can see that there could be an A, T, C, G all randomly present, and this terminal deoxynucleotide can add the things here, and what we will get in final that this ACT was a parental molecule, and this could be addition of the things at the 3' end. This is very important in the level of detection, and also level of doing a mutational thing in the this phenomena of TDT has been used for multiple molecular biology processes such as rapid amplification of cDNA, which is called RACE. That's again a very specific molecular biology process where the TDT can be used in order to add a very specific nucleotide at the 3' end. Now, what's the beauty about this whole process is once you have an unknown sequence, you can tag the sequence. Suppose this DNA sequence, we don't know the sequence at the end of the 3' end. We could do addition of some nucleotide at the 3' end using the TDT. So there could be a possibility that we can just add TTTT at the end of the 3' end. So what will happen once we do the sequencing, we can understand that we have added a TTT at the end of the 3' end with the help of this enzyme. So once this is truncating, we could say that these are the sequence of the gene at the 3' end. So this can give us an indication that what is the exact nucleotide sequence at the 3' end. So this is very important technique called RACE, rapid amplification of cDNA. So this is there. But what is the use of this enzyme and the level of antibodies is we could understand by again going to the same figures of light and heavy chain. So let's, for example, here the light chain. Now since this is a DNA, can we say that this is particularly active here, because this is a DNA. So DNA is only in this state, and after that we are having an RNA after transcription. So this enzyme will be active here. Now what could be a possibility that once there is a DJ or VDJ recombination that can happen in the immunoglobulin chain, this TDT comes into picture and can incorporate few nucleotide into this region of addition. So these TDT can add, so for example V is there, and then here it can add say some other nucleotide in between B and J. So if there is an addition of any specific nucleotide in between the V and J, it can create a mutation. It can create a variation in addition to that. So what is important here is we know that there is already variation happening because of the V and J recombination. In addition to that recombination, the DNA specifically are added with some specific additional nucleotide in between the two segments, and therefore this could lead to variation. And this variation is what is happened because of this enzyme, and therefore this is very critical to understand that this enzyme can do lot of variation in the immunoglobulin chain after the gene rearrangement of VDJ recombination. So this is the second important step in the antibody diversity, where the enzyme can play a very important role in order to do some changes in the sequence of the protein. And finally, we could learn this term called somatic hypermutation. As I said earlier, somatic stands for cell, hypermutation means more mutation. So let's read few of the things and then I will try to explain you. So during the course of an infection, B cell can alter the specificity of antibody that produce, which we have understood clearly that once there is an infection, the B cell can do a specific antibody generation. Mature B cell meet an antigen that it's through its B cell receptor. So what we have understood that basically this is a B cell. So we have the receptor of B cell, which is immunoglobulin, that can bind to an antigen, and So we know that there is a mutation happened because of gene rearrangement. We also know that there is a TDT involvement to do a second level of variation. The third level of variation comes when we have the second enzyme playing a role, and that enzyme is called activation induced cytidine deaminase. Activation is happening because of entry of an antigen, and what it induces? It induces an enzyme cytidine deaminase. What cytidine deaminase does is, if you critically look into, it can do cytidine deamination. It can deaminate, it can remove the amine group from a cytidine residue to make it as a uridine. So cytidine molecules are converted into uridine with the help of removal of this amine that's done by this enzyme called cytidine deaminase. Again, very, very important enzyme that does a lot of mutation in the gene sequences. But here it is very important to say or see why it is happening in the B cell level. Now, just see that we could have a VNC, variable chain, and the constant chain in this immunoglobulin level, and this could bind to an antigen. Now, because there is very light binding, we could see here that this somatic mutation can happen, and this could lead to selection of very high level and high specific B cell population which can bind to the antigen very effectively. So these are the variation which can again happen in the level of B cell, in addition to the rearrangement which we have studied earlier. So this somatic hypermutation can lead to the variation in the immunoglobulin chain, which can lead to production of a very high affinity antibody that can bind very specifically to antigen, whereas a low affinity antibody can not bind that efficiently with the antigen. And therefore, what the body system does is, body system can throw these low affinity antibody producing B cell out and keep this high affinity antibody producing B cell in. And that is what we could see, that there is a selection, and this selection is based on somatic hypermutation, that the B cell which can generate a very high affinity antibody can survive, and the other which can produce a very low affinity antibody can just obsolete from the system by apoptosis or by other cellular processes. So, so basically what is happening is exactly in this way. So the very low affinity, the affinity is low, so the antigen is not able to bind here. So probably this B cell won't survive, while the one which is having a very high affinity can survive and also will be stimulated by this T cell very efficiently. So that means these high affinity B cell could survive into the system, and that is what is understood by the somatic hypermutation. So this is a very, again, a critical phenomena that's happening in the B cell. So till now what we understood is there is a very clear-cut variation in the immunoglobulin chain, and this could happen because of the gene arrangement or because of two enzymes which we have discussed. One is terminal deoxynucleotide transferase, or there could be a possibility of cytidine deaminase. We will see a little more on why this cytidine deaminase is important. I'll just try to explain the same thing in a more elaborative way in the next slide. But these three processes can lead to a very high affinity B cell population that can bind very efficiently to an antigen and just clear this antigen. And this is what this whole chapter is named for, the antibody diversity, how these are generated in order to clear the antigen from the human body. So this is the final slide for today's class. So this is what we have discussed, this enzyme cytidine deaminase. So what it does is, here you have a cytosine residue, and we know that cytosine can bind to guanosine by 3 hydrogen bond in DNA, and if there is a deaminase activity, we see that the amine group is out, and this cytosine is now converted into uracil. And if this uracil is there, this uracil is not at all compatible with guanosine, and therefore in further round of DNA replication, this uracil can bind to adenine, which is here. So we can say that there is a deamination process which can convert the CG to UG, where cytosine is converted into uracil, and further in the level of replication these uracil can bind to A. So ultimately what we are having is actually C to A mutation. So cytosine can cause mutation into uracil, which further lead to variation in the adenosine in the chain, and therefore we could understand that there is a variation in the DNA sequence, and there could be possibility. So what we understood that there is a mutation, and this mutation can lead to a conversion of cytosine to uracil, and in one chain, which could ultimately lead to conversion of uracil into A at the time of DNA replication, which further do a replication where we can see that instead of G and C, now we are getting A and T. So this is a very unique form of mutation which we will see in the DNA, where guanosine will convert into thymidine. Now this mutation can lead to variation in the chain structure, and these mutations are very widely reported in molecular biology. So this is all about the gene rearrangement and immunoglobulin diversity with all the things which we know till date by some enzyme and some of the rearrangement in the B cell. So we will try to understand few more details of this antigen antibody diversity, and how this another molecule which we will discuss, major histocompatibility complex, can bind and present the antigen very efficiently in our next class. Thank you.